So whenever you're going to make a medium formula, you'll need to take into consideration some ingredients. And you have to think about carbohydrates, nitrogen, minerals, and vitamins. So let's take carbohydrates, for example. So as I told you before, fungi degrade or break down polysaccharides into simple sugars. So what you see here, it's the same thing. This is a polysaccharide, and it's broken down by the extracellular enzymes that fungi secrete into simple sugars like glucose. Glucose is also known as dextrose, so probably you'll see in mycology labs they're working a lot with jars like this on which is written quite big enough dextrose. Other carbohydrate sources you'll find in potatoes, agar, and other kind of roots and tomatoes and plants and so on. It's a long list. And as I told you, the other compound should be nitrogen. You'll also find in the mycology labs nitrogen in as a compound like ammonium nitrate or sodium nitrate. So these are compounds that come in jars like these. Now you should use nitrogen, but it's not a rule. In some cases, mycologists, they don't use necessarily nitrogen. They can use only carbohydrates, and of course most fungi will grow on it. But in some cases you have to consider nitrogen as well, because some fungi might not grow on a medium that contains only carbohydrates or nitrogen in very, very low amounts. Glucose, it's responsible mostly for the structure and energy, provides the fungus energy, while the nitrogen, it's going to play a role in protein synthesis and also in enzyme buildup for the fungus. Here I'm presenting you some of the most important minerals important for mycelium development. So you'll need to consider these as important ingredients in your formulation. If your formulation that you're making for a desired fungus and the fungus doesn't grow well on it, then you should also look into adding this kind of ingredients into the formula, like magnesium sulfate or dipotassium phosphate or monopotassium phosphate. As you may see, for example, in magnesium sulfate, sulfur, it's one of the ingredients, and you can find it in amino acids, in cysteine, methionine, thiamine, but also in vitamin B7 as compound there. Now magnesium will play a key role in ATP metabolism, and that's responsible for energy, providing the fungus with energy, and in enzyme activation. And of course the trace elements that you can use in your formulations might be different compounds as sources of iron or zinc or potassium phosphate. It's used in formulation just because phosphate. It's part of some of the important compounds that allow the fungal cell to develop and grow. So for example, it's part of phospholipids. Phospholipids are major compounds of the cellular wall, and nucleotides are the building blocks of DNA and RNA. And you may see here that it's part also of ATP, which is responsible with energy, provides energy in fungi. According to Mas and Chang in 2004, thiamine and biotin are considered to be some of the most important vitamins required by fungi. Thiamine is also known as vitamin B1, and it may be found in seeds, cereals, veggies. While biotin, which is known as B7, vitamin B7, it's found in brewer's yeast, soy, beef liver, peas, and some other ingredients. And also, you may say that also niacin or B3, it's playing a good role in fungus mycelium development, and you can find it in these ingredients right here. Some other vitamins important are vitamin B5 and PABA, or para-aminobenzoic acid. But from all of these, mostly thiamine and biotin will be really important for fungus development. So let's see the next slide, and I would like to show you here 4 media types that are widely used among the mycological society. And as you may see here, the last medium type contains yeast and brewer's yeast. Here in this table you may see how rich brewer's yeast is in a lot of minerals, contains a lot of minerals. And best is if you're going to use the concentrated form of the brewer's yeast, which is yeast extract. And here in these graphs you can see how rich the brewer's yeast is in protein, in fiber, and for example here we're talking in the other slides that potassium and phosphorus play a major role in fungus development. So you can see that brewer's yeast is really high in these 2 compounds. It's not a rich source of carbohydrates or magnesium, sorry, but you may see here that agar has more magnesium than some other ingredients. So a combination between brewer's yeast and agar would be an ideal medium to use. Also here you can see that brewer's yeast, it's very rich in thiamine and also niacin, which we already discussed already. So I believe that yeast extract agar, it's one of the best mediums that you can use in your lab. Also consider this medium. Also consider this medium if you see that your fungus doesn't grow on the medium that you already made.